Hello everyone, welcome back to another G4 Lab IV Biology video. In this video, we'll be covering the basics of the cell membrane, its structures, fluidity, and the components within them. This should tie in nicely with the biochemistry part 1 video that I have made earlier, and hopefully this will make your understanding of the cell membrane far better. Please make sure to watch the first IV Biology video first to get a basic understanding of what the cell membrane is first. Alright, let's cover the first topic, which is the phospholipid. Now, the phospholipid is a lipid that makes up the structures of the cell membrane. So I'm sure you've all seen a picture of a cell membrane before, and in this case, those circle things with the two lines, that is the phospholipid in the visual. And each membrane has hundreds and thousands of these, and these are made in the smooth endoplasmic reticulum, and are transported through the vesicles into the cell membrane through exocytosis, because the vesicles fuse with the cell membrane and increase the overall cell volume and surface area as a result. Now let's look into a deeper components of the phospholipid. Now as you can see, this is a pretty generalized view of it. The actual phospholipid would be made out of chemicals, but in this case, this visual represents a simplified version. Now first, the red circle here is not really a circle, but it is a phosphate head that's combined with glycerol, and this part of the phospholipid is polar and hence hydrophilic. The other two strands are two different fatty acids that are attached to the head. These two are non-polar and are hence hydrophobic. This makes a phospholipid amphipathic, which means they're both loving and hating of water. Now, this amphipathic property is very vital to the double membrane nature of the cell membrane, as it forces the hydrophilic side to point towards water and the hydrophobic side to point against the water. Hence, it creates this double membrane structure that I've modeled here with the visual, in which the tails face inward and the heads always face towards the aqueous nature around them, in which case both directions do have water around them because of the inside of the cell being of cytoplasm and generally extracellular fluid being outside of the cell. Now, breaking down the fatty acids further, there are two different types of fatty acids, one being the saturated and the other being unsaturated. The saturated fatty acids are the straight ones with no carbon-to-carbon double bonds, and they reduce the fluidity and permeability of the membranes, and they also have a higher melting point than the unsaturated, as they are more packed tightly together. On the contrary, we have the unsaturated, which is the bent ones with CC double bonds. They're the ones that increase membrane fluidity and permeability, and on the contrast to the saturated, they have a lower melting point as they are more loosely packed together. Alright, now let's move on to our second topic, which should transition nicely from the fatty acids, of membrane fluidity. Now, again, as I just said, fatty acids have different properties in terms of fluidity, and the proportions in which each type of fatty acids can differ between the cells. Now firstly, longer fatty acids in general will decrease fluidity as there is more friction between a greater surface area. It'll interact more and cause it to be more stiff. And as stated earlier, the type of fatty acid can also affect fluidity. As a result, organisms that live more in warmer climates will definitely have a greater proportion of saturated fatty acids, because those are more tight and you don't want the membrane to be too fluid, to ensure that their membranes stay intact and their overall structure stays intact as a result. Differences in proportion can also be seen in just certain parts of the organism instead of spanning the entire one. Another thing that can manipulate the cell membrane's fluidity is cholesterol. You might have heard about it in foods, but in the case of the cell membrane, it's a little bit different. Now, cholesterol, like phospholipid, is another lipid that is part of the cell membrane and is also amphipathic. Its core function is to maintain the fluidity of the cell membrane, actually, based on the surrounding environment, and it can change the fluidity as a result. At higher temperatures, cholesterol restrains the movement of fatty acids, like it pulls it together, and at lower temperatures, cholesterol prevents the tight packing of fatty acid chains and makes it more fluid so that it doesn't become fully rigid. And now, let's get on to the main show. The topic third for this video is the membrane components. Now, first of all, let's talk about the membrane itself. If you remember from the first video, the purpose of a cell membrane is to maintain and control what goes in and outside of the cell, and kind of create a barrier between what is outside and inside. And if it's the case of a plant cell, there's also a cell wall, of course. But as said before, the membrane is a double layer membrane that is made up of phospholipids and follows the fluid mosaic model. Now, the fluid mosaic model states that the components of this cell membrane and the cell membrane itself is able to move laterally and freely, and are made up of many lipids, proteins, and carbohydrates. So far, I've covered the phospholipid and cholesterol, which are lipids. I will not be covering carbohydrates here except for glycoproteins somewhat, but the main thing to be focused on here is the integral and peripheral proteins that are part of the cell membrane. Now let's talk about the integral versus peripheral proteins. These are both types of membrane proteins. Key difference being that integral proteins are the ones that span across the entire cell membrane between all phospholipids, and common ones of integral proteins would be transport proteins. Obviously, they need to move things in and out of the cell. But on the contrary, peripheral proteins are ones associated on the surface of the membrane rather than spanning across the entire one. And this can mainly be seen through the anchorage and adhesion. Now let's talk about the specific types of proteins that you can find in here. Let's talk about receptors first. There's two different receptor proteins, which are chemoreceptors and sensory receptors. Chemoreceptors are integral proteins that detect chemical signals that can bind to the binding sites that are on the chemoreceptors, like the one in the visual there. And then it can send signals out into the cell to perform actions. And a common example of a chemoreceptor would be the GPCR. The sensory receptor, however, is more a response and a signal, or a receiver to non-chemical stimuli that can close and open chemical channels, ion channels, or send messages to the central nervous system. For example, there are baroreceptors that can detect pressure changes in the cell. Now let's get into transport proteins. Two different types again. There's channels and pumps. The channel proteins allow for passive diffusion of material across the membrane into and out of the cell. You could see this with the potassium channel that can passively allow potassium ions into the axons of neurons during action potential repolarization. For pumps proteins, however, these are the ones that actively move things in and out of the cell membrane. The difference between the channel and the pump is that the pump is active and cannot allow passive diffusion and requires energy to, quite literally, pump things in and out. For example, the sodium-potassium pump actively moves sodium ions out and moves a few potassiums inside to the cell. And now let's get into the anchoring and positioning proteins that can help the cell. First, we have the adhesion type of proteins, in which these are the ones that allow cells to form tissues. If you remember, tissues are made up of a group of cells specialized to do the same task. So through these adhesion proteins, cells are able to form tissues by adhering to neighboring cells. They're made out of cell adhesion molecules, and these CAMs, as they call it, are embedded within the plasma membrane to bind cells with other cells, or even with the ECM, which is the extracellular mix. However, more for the ECM is the anchorage proteins that allow cells to anchor onto these ECMs. And this allows the cells to have support, and if it doesn't have any other cells nearby, it allows it to anchor in place. Finally, we have recognition and enzyme. These two are not related. These are two different types of proteins. But recognition is a protein, or recognition proteins are proteins that can help identify different interactions or materials that are around the membrane and cell. Often glycoproteins that are a type of recognition have carbohydrates attached to them. And lastly, enzyme. You've probably heard of enzymes before and their function of catalyzing, which means speeding up reactions for various metabolic purposes. An example of this would be the ATP synthase, which can create an energy storage molecule called ATP during cellular respiration and photosynthesis, which you've probably also heard of before. Now let's discuss a little more about the recognition protein, the one that IB would want you to know, which is the ABO glycoprotein. So depending on the glycoproteins that are on the blood, it can determine what type of blood can be fused together. For example, the O type can be fused with all blood, A can be fused with A and AB, B can be fused with B and AB can only be fused with AB. The thing that helps find this is these recognition proteins called the glycoproteins on the blood cells. Because of this property, these ABO glycoproteins are known as antigens. And that is it for the membrane structures unit of G4 Lab's IB Biology. This will be the last video that I will be making, and hopefully this project will be passed down to other students so that we can provide you with more information on the IB Biology exam. Please make sure to study hard as the exams are very soon, and we hope to see you in another video sometime soon, whether it is in biology, computer science, physics, or maybe even more IB subjects.